BU Wellesley software team, and the three of us who are going to be presenting today are representing the three different subunits of our team. I'll be talking about the human-computer interaction side, including tools like GenomeSurfer Pro and Optimus Prime.
Hi, I'm Margot. I represent the wet lab of our team. We had two goals this summer. One was to work towards a synthetic biology problem, and the other was to use the tools that both of our computational teams created for us.
Hey, I'm Jen Hong, and I'll be representing the algorithmic back-end side of the team.
So when we began this summer, we looked at the challenges that face synthetic biologists in the research that they have to do on a day-to-day basis. And we found that these challenges fell into three main areas: the management, organization, and relation of complex data, the design of biological systems, and the deployment of actually creating those biological systems in the wet lab. Now, there are lots of subproblems that arise in each of these areas. And from those subproblems, we identified three key areas that we thought we could insert computation in order to make synthetic biologists' research easier. And those three areas were the characterization of biological systems, assisting collaboration between wet lab users, and dealing with the complexity of the systems that they're working with. So our vision from this information for the summer is that we wanted to apply engineering principles to synthetic biology by utilizing user-centered design to build software tools that foster collaborative problem solving, support complex system design, and enable automation in the lab. Now, looking at all of those problems, it's difficult to imagine that a single software tool would be able to help with all of those challenges that researchers face. So we created 5 tools in a complete workflow that takes synthetic biology researchers through the entire research process. Those tools are GenomeSurfer Pro, Optimus Primer, Trumpet, Puppet Show, and the Electronic Lab Notebook. Having created these tools, we also tested them out with our BU Wet Lab team to make sure that they were actually useful in the lab. So coming from human-computer interaction, there's a technique that we use called user-centered design. And the basic idea behind user-centered design is that users should be involved in all aspects of the design process. Now that seems obvious. The people who are going to be using your system should be involved in helping create it. But for us, there's a lot that goes into that. We start with background information, doing background interviews with synthetic biology researchers and observing them in the wet lab to identify what challenges they face. Afterwards, we create preliminary designs of systems that we think can help address some of these challenges. And then we bring those designs back to the wet lab users, asking them for feedback on what we thought and iterating on those designs several times before we create any computational product. The idea is that after doing all of this iteration, we know that we're creating something that will be desirable, usable, and useful to the actual researchers. So I told you that the first step in this process is doing background user studies. And this summer we interviewed 32 researchers about their synthetic biology work. We conducted 1-hour interviews in their laboratories and asked them questions about the software tools that they used in doing their research, about their typical workflow while working through a research question, and to walk us through one of their research questions that they were working on, from the beginning of hypothesis forming through the actual work that they did in the lab. And from all of this, we were hoping to come up with some design requirements for any software tools that we would build. We were hoping to find things that lots of synthetic biology researchers found difficult so that we could help make those easier. And afterwards, we did find several trends, several requirements for any software tools that we would create. And those requirements were that our software tools would help to alleviate data explosion, integrate the workflow by allowing you to use multiple tools within the same application without having to switch between applications, provide various forms of information by integrating tools that allow you to compare different forms of information to each other, to support users from novice to expert, so to support novice users even if they don't have a huge amount of synthetic biology research experience or experience with computational tools, and to facilitate collaboration between wet lab users. So now that I've told you about the requirements that we created for any software tools that we would build, I'll start telling you about the software tools that we actually built to fill those requirements. And the first one that I'll talk about is GenomeSurfer Pro. And GenomeSurfer Pro is a tabletop interface for the collaborative exploration of genomic data. And the idea is that you could use this while forming a hypothesis at the beginning of a research project. So if you have a bunch of genes that you are interested in, then on GenomeSurfer Pro you can pull up information about these genes, including their sequence and translation information, as well as PubMed publications about that gene, and you can blast that gene against the entire NCBI database. And since all of these tools are united in one system, you can then relay that information to each other spatially on the surface. This is a quote from Natalie Koldell of MIT and the Synthetic Biology Engineering Research Center that illustrates why such a system could be useful to synthetic biology researchers. So another tool that we created this summer was Optimus Primer, a primer designer. Now, I know that you've seen a lot of primer designers come through iGEM before, but there are two things that make our primer designer special. The first one is that it interacts with Clotho, meaning that any primer that you design, you can save to your Clotho database. And this allows you to reuse that primer, to send that primer to other researchers in your lab, and to share it across wet labs to allow for greater collaboration. The other exciting thing about our primer designer is that it's integrated with GenomeSurfer Pro on the Microsoft Surface. And this is nice not only because it integrates these different tools into one platform, but also because learning primer design is difficult. Primer design isn't something that intuitively makes a ton of sense, especially when you're new to synthetic biology research. Traditionally, when you want to learn how to do primer design, you as a new synthetic biology researcher are watching an instructor or an advisor as they go through the process. So you're watching as they use a tool on their computer or as they design a primer by hand. We think that this watching to learn is not as useful as actually learning by doing. And by implementing something on the surface, you can get multiple people sitting around actually getting their hands on the data and learning by doing. So at the beginning of the summer, we started with a low-fidelity paper prototype of what we thought a visualization for prokaryotic genomic data might look like. And starting from just this paper prototype, we built from the ground up a complete system that addresses all of these challenges. Here's our system. So after we had created this system, we needed to evaluate it to check that it was actually useful to wet lab users. And we conducted a 3-tier evaluation to go through this process. We did a usability study in which we brought researchers into our HCI lab to test the concrete interaction techniques and visualizations that we had created. In the future, we hope to do an impact study in which we can evaluate the usefulness of GenomeSurfer Pro in an end-to-end research workflow. The evaluation that I want to focus on in this presentation is the usefulness evaluation that we conducted both with an intermediate neuroscience laboratory at Wellesley College and also with our synthetic biology wet lab team at BU. So we brought this tool into their labs and let them solve a research question that they already had. Let them use the system to do something that they were already familiar with. And we observed them and gave them questionnaires to obtain some concrete results from this information. So we used the NASA TLX questionnaire, which is a standard measure of workload, as well as a standard engagement questionnaire, to obtain these numerical results about people's experience with workload and engagement. And from this we can see that users had very high confidence in the work that they were doing with our system, as well as that they enjoyed it very much. We also used a standard series of metrics for measuring the different dimensions of collaborative learning. And from this we can see that there's lots of collaborative learning happening in lots of different dimensions. So overall, our findings from these studies were that GenomeSurfer Pro reduces the workload, improves collaboration, encourages reflection, and facilitates intuitive interaction. So after we had done all of this evaluation, we released GenomeSurfer Pro and Optimus Primer for use by our wet lab.
So as I previously mentioned, our wet lab had 2 main goals. One was working towards a synthetic biology problem, and the other was using these tools to do so. The synthetic biology problem that we looked into was studying the genetic regulatory network of tuberculosis. So we used GenomeSurfer Pro to go through and select what genes of this network we wanted to study, and they are listed on the slide. And subsequently, we had to then design the primers and biobrick them. And built into GenomeSurfer Pro is Optimus Primer, a primer designer, and you can see on the slide as well the primers that we created.
As Margot previously mentioned, this summer we were interested in investigating the interaction of genes in tuberculosis. Shown behind me is a gene regulatory network of tuberculosis. We wanted to investigate this complexity without constructing a huge number of parts. Yet at the same time, we want to create sophisticated genetic circuits, not just for exploring tuberculosis, but also for other novel applications. Such genetic circuits should capture a large state space, which can best capture the complexity of our desired applications. This summer, our solution to these two challenges is a software tool that we call Trumpet, which is a play on the word permute. We started this summer by asking, can we make a reconfigurable biological construct? Previous work has been done in this area. The Kiesling group made a 5-state machine using invertase, and the Collins group, using invertase as input, made a 3-state counter. However, it is not clear whether or not we can create a fully permutable construct. By fully permutable, I mean given a set of parts, say A, B, and C, we should be able to achieve any permutation, BAC, CAB, and so forth. After a summer's worth of work, we developed 2 algorithms for implementing such designs. But before going on to discuss these algorithms, let me discuss invertase with you, which we use to reconfigure our construct. Shown here are 2 triangles which represent invertase recognition sites. Invertase, which is a subset of recombinase, will recognize these sites and invert the segment of DNA between the sites, like so. This inversion will effectively turn a part on or off. And so up here at the top, we have 4 parts, 1, 2, 3, and 4. This is not the permutation I wanted. I actually want 2, 4, 1, and 3. To achieve my desired permutation, I can use one of our 2 algorithms, link sort or pancake. In the interest of time, I'll only be discussing pancake today. Pancake uses 2 operations, bring to head and push into place. Watch carefully part number 3 over here. We're going to bring it to the head using an inversion, again using invertase. 3 is now at the start, and all the parts that have been inverted, 1, 2, and 3, now have different orientations. We're now going to push 3 into place at the very end. And so again, we're going to invert all the parts. 3 is now in its final place, and again, all the pieces now have different orientations. We're going to repeat these operations on the rest of the parts. So again, bring to head, push into place. We now have our desired permutation, 2, 4, 1, 3. We're going to do one last operation to give our parts a proper orientation. And there we have it, our final desired construct with proper orientation and order. The complexity of our designs depends on 2 factors, the number of inversion operations and the number of invertase sites. The pancake algorithm, which uses nested invertase sites, needs 1 recognition site per part for a bring-to-head operation. And for each push into place operation, it needs n sites per part. Shown here are 2 designs. At the top, we have a design generated using link sort, and at the bottom, we have a design using pancake. The letters denote parts, and the triangles denote invertase recognition sites. As you can see, even with a small number of parts, the designs are intricate and complex. I would like to take this moment to ask those of you in the audience how one would conceive of such a design. To avoid an uncomfortable silence, I'm just going to refer you to our software tool. Shown here on the left is a design we've proposed that's meant for novice users, where one would drag your parts of interest into this bin, click a button, and the Trumpet algorithm will take care of the rest, generating designs using either the pancake algorithm or the link sort algorithm. On the right here is a desktop photo tool, which is fully implemented. Again, drag your parts of interest into a bin, hit a button, let Trumpet take care of the rest for you.
job explaining how Trumpet has all these algorithms and how they work. But biologists, what does this really mean to you, this output of Trumpet? Well, if you see up here, P1 and P2 correspond to two genes of interest, and these numbers 3, 2, and 1 refer to these different invertases over here and their corresponding sites. If I flank my genes with these sites and then add, say, one of these invertases flip, what will happen between the two pink arrows here is the genes will be flipped and inverted, effectively turning them both off. We can use this kind of idea to go through and study the interactions of the different genes by turning one off and one on. And you can also see from this example that using two genes and three invertases, just how complicated this outcome can be.
Alright, so now we have a design that we made using Trumpet, and now we want to build a physical construct. We might want to use a liquid handling robot to avoid all the manual labor ourselves. There's one problem. Using current software to create a 10-part bow brick, it will take about 200 lines of code. To avoid all this complexity, this summer we designed Puppet Show, which is an automated workflow. And so you can make that same 10-part bow brick using just 20 lines of code. Puppet Show implements the following workflow. We define protocols, call these protocols like we do functions, which sends instructions to the robot. I would now like to point out some of the key features of Puppet Show. Puppet Show is an interface for defining protocols and specifying assembly programs. It will automatically generate Puppeteer code. Puppeteer is a high-level language for specifying biological protocols. Puppet Show automatically optimizes labware allocation, and so you don't need to worry about where wells and plates are. You can just specify the sample that you want to use. Each of these samples are saved, as well as its entire protocol history. And so you can look at each sample and know exactly what protocols was operated on that particular sample. For those of you uncomfortable with writing your own Puppeteer protocol, we've made a Puppeteer protocol repository. For protocols such as BioBricks, which we've implemented this summer, we're in the middle of implementing a Gibson-based assembly method. We hope this repository will be a medium of exchange among the synthetic biology community. So again, here's the workflow. We start with a design that we can grab from software like Trumpet. Puppet Show will arrange these into a set of sub-cloning operations, which can be implemented using a protocol like BioBrick. Puppet Show will generate this Puppeteer code automatically, which looks complicated, but the base robot instructions are even worse. Again, none of this is something you have to deal with. Puppet Show does all of this automatically, and so this summer, with Puppet Show, we were able to make constructs, verify them successfully in our web lab.
The electronic lab notebook is another tool that we designed this summer. It came straight out of our conversations with synthetic biologists about tools they would like to see in the lab. Now, the electronic lab notebook is something that we plan to fully implement and test in the wet lab next summer. So in order not to spoil the surprise, I will refer you to our wiki if you have any questions about this tool.
To have another method available to us to create our constructs other than by hand. You can see here an example of a protocol we would do in the lab in our actual notebooks, versus here is the output of a code from Puppet Show corresponding to the same protocol. Like Jen said, we went through and we verified it, and in this gel image here you can see an uncut BioBrick, and then we went through and we did restriction digest by hand, which is shown in column 2, and then columns 3 through 5 showing the results of restriction digest done with the robot, and you can see that there is no obvious difference.
So, in the interest of time, I think we'll breeze past future work. We covered that earlier. To just mention that we think that this system that we've created this summer will have a very large impact on the synthetic biology field. We've shown that it's feasible to streamline the entire experimental process into one suite of tools that supports novice users and collaboration in a wet lab setting, and we successfully tested these tools. And here's a quote from George Church, who we interviewed at the beginning of the summer, that highlights the usefulness of the sort of research that we've done this summer. Thank you for your attention, and we would love to take your questions.
So maybe my first question would be to have you detail a little more how you organize your work. Did you split into different projects, and which project was the most time consuming?
Actually, I would think you guys would go first. So we in the wet lab set out to try and first create the fluorescent protein devices to be fused with tuberculosis genes. And then as the tools became available to us, we went back and we kind of used them in our process. And we then started looking at the tuberculosis genes when we used GenomeSurfer Pro. And then, as we're still working towards creating our constructs with the configurable invertases, and we'll be looking to trumpet at that time. And we also used Optimus Prime to create our primers. And I think I'll send this back to you guys.
So from the computational side of things, we've had a couple of projects that we worked on across the sort of the BU Wellesley gap. We didn't have too much of a gap. We were working on most of the same projects all summer. So the Optimus Primer and Trumpet were things that we worked on between the two teams collaboratively. GenomeSurfer Pro was something that was implemented pretty much entirely by the Wellesley team, and Puppet Show was implemented pretty much entirely by the BU team. Is that fair?
Internally, we also communicate a lot. So while I didn't work directly on Trumpet, here I am presenting it today. I talked with the wet lab almost every week to optimize Optimus Primer for their convenience.
And I'll also mention that as part of our research, the background research that we did into how synthetic biologists use tools, Wellesley was in the wet lab at least once a week all summer. At the beginning of the summer, more like 2 or 3 times.
What was the main gap and challenge between the web lab team and software team?
I guess one of the first big gaps we had was trying to get everybody on the same page in regards to biology and computational terms and all that kind of background. And so we all had to very quickly learn how to disseminate really tough concepts from each of our backgrounds, and at the same time also learn how to take this disseminated information in order to go and work off of that. Does that answer the question?
And what about just the software team?
For us, the hardest thing, I haven't taken biology since my freshman year of high school, so I had a lot to learn. We had a 3-day boot camp at the beginning of the summer to sort of catch all of us up to be on the same page in terms of biology and in terms of the computational tools. And then every time that we had questions, we could go ask the wet lab. So that was the biggest gap, but we bridged it very effectively.
Well, as I know, there are many tools available out there already. For example, for primer design, do you know there are tools available already for genome browsing? There are a lot of tools available for genome browsing. For assembly, like DNA Designer can do assembly already. I didn't see any comparison you have here with those tools. So how do you know your tools are useful compared to those tools? Do you have any advantages?
And so we didn't discuss those other tools like you've mentioned, but we are fully aware of them. A lot of the existing tools do implement what our tools have done. What our tool does is that it leverages Clotho. And so Clotho, as you have seen in previous iGEM competitions, is an open source software platform. The heart of Clotho is a common data model and a plugin infrastructure. And so while working with existing tools out there that we didn't discuss today, you can piece them together, but you have to copy and paste results from one tool to another. Using Clotho to unify our tools, the transition is seamless, and so we can enable easier interaction with the data via drag and drop and so forth.
So before we designed the tools for the summer, I did a lot of background research, as well as with the rest of the HCI lab. We did a lot of background research on available software such as CG Viewer and APE and Gene Designer and also Genius. And then we looked at those tools and then looked at the pros and cons of each tool and then used that to inspire our design. And then we thought it would be really interesting to compare those tools to our tool. but that our tool has an advantage in that there's a lot more surface real estate on our tool, and then we can use it for education and collaboration, as opposed to Genius, which is a lot more expensive and very difficult. It's a very high-end tool, so the threshold for learning is very high, as opposed to this, which is much easier. And so if you want to teach somebody such as like Primer Design or Gene Browser in a just like beginning level bio class, then you can use this tool instead.
Okay, next question.
Okay, so outstanding work actually. So I will continue with the previous question concerning the Gene Browser. What kind of database behind? So you can integrate different workflow. So what kind of workflow you can integrate?
So right now we're getting, we're generating the gene visualization from the GenBank file. So you're getting all of that information about sequences and translation, and as well as the GenBank notes. We're also providing live articles from PubMed for a specific gene. So you're getting the sort of very basic information about the gene as well as the research about the gene. And then from there you can move directly into your primer design process. And from there we have algorithms about the primer design and also things like the NCGI BLAST, where we're making use of their databases.
And again, I'd like to point out that the surface applications are obviously not Clotho tools being implemented on another platform altogether. We'll hold our two different platforms together. The desktop and the Microsoft Surface is a Clotho data model. And so the Surface application for Optimus Primer communicates to the same database as the Clotho implementation. And so that way data can be exchanged between the two sites.
Okay, so we need to move on, but thank you.